DIAKEY REALcheck Viral DNA and RNA Prep Kit. DIAKEY REALcheck Viral DNA and RNA Prep Kit is able to be used with DIAKEY REALcheck COVID-19 Detection Kit. DIAKEY REALcheck Viral DNA and RNA Prep Kit for column type. The kit is prepared to perform 100 individual tests. Components. This includes the quick guide and proteinase K, VW1 washing buffer, empty bottle for VW2 washing buffer, lysis buffer VL, RNase-free water for elution, capsule column, cell lysis. Add 150 microliter sample to prepare 250 microliter VL. It is optional, but we recommend to add 2 mercaptoethanol to get high-quality DNA or RNA. In case of enveloped virus, please incubate the mixture after addition of 20 microliter proteinase K solution. And perform vortexing for 30 seconds to 1 minute. Incubate the mixture for 10 minutes at room temperature. Column binding and washing. Prepare the column on the rack. Add 350 microliter 100% ethanol to lysate. And perform vortexing for 30 seconds to 1 minute. Transfer lysate into the capsule column. Perform centrifugation at 13,000 RPM for 1 minute. Discard the flow-through solution. Column washing. First washing. Add 500 microliter VW1 into the column. Perform centrifugation at 13,000 RPM for 1 minute. Discard the flow-through solution. Second washing. Add 500 microliter VW2 into the column. Perform centrifugation at 13,000 RPM for 1 minute. Discard the flow-through solution. Centrifuge again, at 13,000 RPM for 1 minute to completely remove residual ethanol. Place the column into a new 1.5 ml microtube to collect DNA, or RNA. Viral DNA and RNA elution. Add 50 microliter RNase-free water into the column. And incubate it for 1 minute at room temperature. Perform centrifugation at 13,000 RPM for 1 minute. Remove the column. The eluted viral DNA, or RNA, is in the microtube. Please store at appropriate temperature.